Hello, my name is Armando Hernandez-Garcia. I'm a professor in the National University of Mexico. Today I'm going to talk about my last research, which is focused in building nanostructures combining the application of proteins that bind to DNA and double-strand DNA. As we know, DNA has become a very versatile building block. It has so many applications in the last years, and we hope and we expect to be even more important. So we need more tools to manipulate and control properties and build easier, cheaper nanostructures based on DNA. Currently, there are two main strategies to build these DNA nanostructures. One, which is depicted here on the left, in these red arrows, is based on the nanoparticles that are already rigid. And these rigid nanoparticles are decorated with DNA, and the DNA is forming the bonds among these different nanoparticles, so organizing it and forming the final nanostructure. And the other strategy, which is more common in DNA nanotechnology, is using the DNA by itself to cross and also to rigidify and to form the bonds. So this is the hybridization based. And although these strategies have had very important advances in the last years, and most of the DNA is currently made in this way, I think there are still some limitations. Like if you want to build larger structure, you need more single-stranded DNA strands, and this sometimes became so expensive. And also, this represents that they will have very complicated self-assembly processes, which are sometimes unknown or even uh, impossible to know. So, as we use also in this strategy for the DNA nanotechnology, single-stranded DNA is almost impossible, very, very difficult to synthesize these structures inside cells, which need double-strand DNA. So one of the motivations for my work is how we can build DNA nanostructures which are cheaper and easier. So our main strategy is using protein brushes around DNA. So basically the concept is that we will use a DNA binding protein that could heavily coat single DNA molecules, and in this way we can stretch and stiff these DNA molecules. And we know this pretty well from already polymer science, how these things can work. So with these proteins that coat DNA, we can control physical properties of DNA, and also it can become a way to display other molecules. Um, we know that DNA is a very flexible molecule, especially when it's single-stranded, because its persistent length is around 3 nanometers. When it's double-stranded, it's around 50 nanometers, which is pretty low, so it's very flexible. So because considering that is sometimes we have DNAs of micron size, but with a persistent length 10 times smaller. So in the last years, we have built a family of different proteins, which we know they stiff quite a lot sometimes DNA. For example, this dye block can increase its persistent length the 4 times, this even 6 times, just forming a coating around the DNA, so as you can see here. So I will focus my presentation in these two protein dye blocks that we have already reported before in all these publications. These proteins are designed in a way that they are two blocks. One is for what we call colloidal stability, which is just this sequence based on collagen-like sequence, and we fuse it genetically to a binding domain, which either can be just 12 lysines, so for electrostatic binding of DNA, or this one is a specific domain that derived from the archeal Sulfolobus sulfataricus. It's called Sso7d. It's 63 amino acids. It recognizes specifically double-strand DNA. So we have these two proteins, and we know from already previous reports that they are able to code single molecules of DNA, either linear DNA, you can see how they are coated, but even other more complex molecules in shape, like circular supercoiled DNA. In this case, this Sso7d can coat around the circle, or even the circular but single-stranded DNA. So this is a zoom into the brush. You can see how it's very dense, this brush of the proteins around the DNA. So actually this effect of stretching DNA by coating them with proteins has been already used for many applications. For example, in this one, we have very, very long DNA from T4 fake, which is around 56 microns of contour length. And inside nano channels, we could stretch it just by adding a protein of dye block. And you can see as we add more and more protein, even 2 times the negative charge of the DNA, we can almost stretch it completely to its all contour length. So this actually allows to resolve some fluorescence markers very well inside these nano channels. So another application has been coating origamis of DNA, even 2D or 3D nanostructures. So as you can see here, this rectangle and this kind of pyramid-like structure with one of our proteins, and they didn't destroy it, they didn't affect its integrity, so they are just coating them, and actually they prevent degradation for longer time. So here the idea in this work was to build some nanostars with DNA and coating them with the proteins. So nanostars are very important structures, so they can be used to display different molecules around, and to use it as a building block for hierarchical structures. So they have many applications in materials. But so far, just small stars have built, like a few nanometers. So it's challenging to build larger ones. As I said before, they are expensive. It's a difficult assembly process. It's not impossible, of course. There are so many reports, but we want them to be easier and even cheaper. So our concept was very easy. So we built first a nanostar made up of double-strand DNA arms, and just by adding the protein, we will stretch it and rigidify it. So we have this more regular and well-structured nanostar, DNA protein nanostar. So basically, to build this naked DNA nanostar, we need a junction, a 3-arm junction with 6 sticky ends. And so just mix it with the double-strand DNA that carry sticky ends opposite to these ones here. And then we just coat it with a protein. So we use some already reported DNA junctions, especially from Chang De Mao group. We redesigned the sequences in order to have some particular sticky ends at the end. So especially 6 in these 3 arms, so 2 per arm, by a simple self-assembly process. So we focus in this 3-arm junction, but we can create easily 5, 8, or any other number of arm junctions. So depending, just changing this the sequence of these strands. So after we have to synthesize the double-strand DNA, especially with some overhangs or sticky ends that could match the ones in the DNA junction. So for this, we use chemically modified oligos, which they have a spacer among the sticky end and the primer. And this spacer just make them at the end to be with this area or this part here just a single strand, so they are ready to hybridize with the sticky ends in the junction. So, and this is the way how we build the nanostars, the DNA naked nanostars. So here is an agarose gel where you can see when you mix this double-strand DNA with the overhangs, with the DNA junction, the 6 sticky ends, you can see how these different bands are formed, which indicate the different number of arms in one junction. So we can go easily from 1, of course, to even 6. So here the 6 is not well resolved, but later we confirmed by AFM that it was present. Actually, in the agarose gel, this long of the 6 branches nanostar is kind of destroyed, as we show in this paper. But anyways, here we show that we can self-assemble easily. So actually, when we add this the DNA without the overhang, like in this case, we cannot form any of these nanostars with different arms. So the next step was to coat them with the protein. So for that, we have here some agarose gels, some EMSAs, this test where we can see the change in the mobility of the DNA when we are coating them with the protein. So here at the top indicates the proteins per base pair, or proteins number of positive charges per negative charges of DNA. And as you can see, high protein concentrations or high ratios of protein to DNA, you see a band at the top which indicates that the DNA is being coated. In this case, are the nanostars are being coated by, in this case, by these 2 protein dye blocks. To confirm that we were getting the stars, we look at the AFM, and as you can appreciate, we found quite some of stars, actually with 6 arms, which, as I mentioned before, in an agarose gel they were not really seen, these nanostars with 6 arms, but they are here by AFM pretty well. You can see they are very well organized, they are regular, sometimes they are forming in large numbers, and everything is expected. So one interesting thing is when we try to coat them, when we test different proteins to coat them, the outcome was different. So when we use this protein, the C8Sso7d, which is based in this domain from the archeal Sulfolobus sulfataricus, and it binds specifically double-stranded DNA. So we see that the stars are formed well, are well formed. So, but when we use this other dye block, which relies on binding the DNA just electrostatically with 12 lysines, the stars look a kind of a mess. So like kind of destroyed. And this protein is like this dye block, but it has a self-assembly silk-like domain in between, which just tends to form these rods. So depending which protein you use is the outcome you will get. So you need especially this a protein that binds specifically and non-electrostatically to double-stranded DNA. So here I'm showing more images of AFM images of these nanostars coated with DNA here, sorry, coated with a protein, the naked ones, and here I'm showing some distributions of the number of branches. So see, these ones are on the right are the percentage of branches. Sorry, the percentage of nanostars with 6 branches, 5 branches, etc. And you can see when you have them, especially these short ones, coated with protein, it seems that apparently there are more. So apparently, coating the protein, the DNA stars with the protein, make them a bit more stable, and for that reason, we see them in higher numbers, especially when we use this kind of short arm and not this long one of 7 to 2 base pairs. Here you can see at the top of this AFM image nanostars, DNA nanostars, which are naked, and they were built using different arms of different length. So this one is 70 nanometers, and on the extreme right you can see one with 250 nanometers length arms. So, and at the bottom, when they were coated with the protein, the Sso7d protein, so there is a clear difference. So they look more regular, they look more symmetrical, and of course more rigid, more stiff. And we attribute this to the effect of the protein brush around them. As I mentioned before. So after we wanted to see how stable were these nanostars got coated with the protein to the enzymatic attack. So we just incubate these protein DNA nanostars with the DNase 1 for different times and see the integrity of the band. So depending if it was destroyed, the band will disappear completely or partially. So here in this agarose gel at the top, we just see the arms, so the double-strand DNAs, which are not forming stars. And after 1 minute of incubation with some amount of the enzyme, it's completely gone, which is expected. When we self-assemble these DNAs in a nanostar-like shape, so they were more resistant. All the bands disappear around 5 minutes. So this already suggests that organizing the DNAs in stars give it a bit of more stability against this enzymatic attack. But when we coat them with the protein, as you can see this band here in the well, of course they don't move in the agarose gel because the charge of the DNA is neutralized by the protein, but anyways you can see the band. So this band remains at least here for 60 minutes. So in another gel that we follow the incubation with the enzyme for longer, we see that still the band is a bit visible after 10 hours. So this indicates that nanostars coated with the protein are much more stable against enzymatic attack than naked nanostars or just simple DNA. And what about the thermal stability of the nanostars? So first we measure the melting temperature. Here you can see at the top the fluorescence of the DNA that we follow with an intercalator. So the melting temperature shift to the right when we have DNA coated with a protein, and when this DNA coated with the protein and also in nanostar shape also is shift to the right. So meaning they became more stable. So these protein DNA nanostars become more resistant to melting. And something similar we appreciate by AFM, the structural integrity of the nanostars are preserved when we incubate it, especially at 37 degrees, which is important for biological applications. So after 3.4 hours, you can see some nanostars, and even after 9 hours at 37 degrees, you can still see some nanostars, protein DNA ones. So although it's not completely perfect, they remain. So in summary, we have a way to build these nanostars using a protein that bind to DNA, which are especially coating and stretching and stiffening these DNA nanostars built of double-strand DNA. So we believe this is a new strategy. It's not based on nanoparticles, which are already rigid, or just crossing over many single-stranded DNAs. So it's a completely new strategy. We believe. So it's a easy and cheap way to produce these nanostars of different sizes, which we can program in number of arms, in the length of each arm. And we believe these protein DNA nanostars can be a new, or can be used as a building block for hierarchical materials. And of course, they also have many applications that we are now exploring, especially to display bioactive molecules on the surface with high density. And finally, I want to acknowledge and to thank the Eddie Sanchez, which was a student that made most of the work, and he got his master's degree with this work, and also the funding by the GAPA at UNAM in Mexico, and to all the group. So thank you very much. If you have some questions, you can leave it probably in the comments, and I will do my best to answer them. Thank you.